Ken has been waiting for a little while. I saw you in that waiting room for a little while there, Ken. Welcome. Well, I am going to get us started. Welcome to, it's Friday. We made it. I don't know, it feels like it's Monday still to me. They all kind of blend together. I don't know about the rest of you, but we're really, really excited for our talk today. We have the amazing Dr. Subramanian, who I'm going to let you introduce our speaker, Dr. Subramanian. But just a quick little housekeeping. We will take all of the questions in the chat. So if everybody wants to say maybe where they're from, where we love to hear where around the country or around the world that you are. So all questions will be taken there, and this is being recorded just so everybody is aware, and there's no slides today, so we will not have those available. So Dr. Subramanian, I'm going to kick it over to you just to get us started.
Okay, perfect. So thanks everyone. Great to see everyone joining us, and hello from sunny Southern California. Hopefully everyone's staying safe, and now I guess on top of the pandemic, people are battling some fires in some places, so hopefully everyone's doing okay. So we're still virtually connecting and socially distanced, but we're thinking about all of you and missing seeing some of our patients in our clinics. So enjoy, hosting this. And so it's with great pleasure that I bring you a friend, Raj Pahwa, today, who's taken some time out of his busy schedule to join us. He is kind of a legend in clinical trials, and even Mike Oken, who is, a friend of both of ours, had tweeted out that, Raj is just, sort of the maven of clinical trials, and so we're just excited to have him and to pick his brain a little bit about, some thoughts around how patients can get more involved in trials. Why is it hard to study Parkinson's, and what are the complexities of these trials, and what is he excited about in the future in terms of some of the newer sorts of things that are in trials right now. So we'll sort of learn a little bit about Raj first, and then kind of get to asking some of those questions, and we'll be taking some questions from the chat too, so go ahead and put those in there. So Raj is currently at the University of Kansas Medical Center. He is the Laverne and Joyce Ryder Professor of Neurology and Chief of the Parkinson's and Movement Disorders Division. He's the director of the Parkinson Foundation Center of Excellence, which is a really cool center of excellence that they have out there. He's collaborated over the years with me and just been a great mentor and taught me a lot about how to meander through navigating things in the promotions world and the trials world and the meetings world. And so just been a great asset to have you, Raj, and thank you so much for everything you've done for me. And maybe you can tell us a little bit about what made you interested in medicine, kind of how you got interested in neurology and movement disorders, and maybe more specifically about how you developed a passion for clinical trials.
Thank you for having me, Indu. It's an honor being here with talking to a lot of advocates for Parkinson's, and it is a journey that we have all been through. My interest in being a physician started when I was a kid. As you know, like me, you're an Indian, and our parents want a doctor in the family. And I was the last kid in the family, and none of my other siblings had been a doctor. So right from the beginning, my mom just kept pushing me, okay, you got to be a doctor, you got to be a doctor. So, I didn't have too many other choices, so ended up being a doctor. The next thing, as you know, comes down to is what kind of doctor you're going to do after you finish your medical school. And when I had finished medical school, I did some research in head injuries. So it was kind of a neurological thing, looking at brain, and that kind of got me interested in neurology. So you become a neurologist, and then what's the next thing you're going to do? So my interests have always been treatment part. There are people who like to diagnose things. There are people who like to do tests for certain things. There are people who have different interests as far as even medicine is concerned. And for me, it was always the treatment part. And when you look at neurology, and here we are looking at about 30 years ago, there were not many therapies available for neurological conditions. You had dementia, we had nothing for it. You had epilepsy, yeah, you had a lot of drugs for epilepsy, but then you had to read those EEGs, which are crazy lines on a paper, and all those lines look like as if the computer was not working well, kind of a thing. And then you had neuromuscular things, which again, there was nothing there for them. Stroke, we just used aspirin. So really, MS, again, all we had was steroids. So there were really no treatment, except for Parkinson's. We had a few drugs. We had levodopa, we had dopamine agonists. We even had MAOB inhibitors at that time. Of course, we had amantadine. So at least we had a group of medicines available when I was finishing my neurology residency. And I was like, okay, at least I have people that I can treat and make them better and hopefully manage their medical issues. So that was my interest in helping people getting better, rather than saying, okay, you have Parkinson's and there's nothing I can do for you. Well, there was still a lot we could do for Parkinson's even 30 years ago. So that's what kind of got me interested into Parkinson's. And the clinical trials basically started with my mentor, because when I did my fellowship, my mentor at that time, Bill Kohler, he used to do a lot of clinical trials like I do today. And right from the first day I did my residency, I started doing clinical trials. In fact, during my fellowship, my first clinical trial was on Cinemet CR. And that was my first big publication that was out there. At that time, you don't realize how big this publication is in the Green Neurology Journal. You being a first author, you think, oh, that's something you can do anytime. But then you learn over your 30 years, well, it's not that easy to get a peer-reviewed publication in that journal. But that's pretty much what started it. That started my road up clinical trials. And in the last 30 years, I have done clinical trials related to pretty much different aspects of Parkinson's disease, and pretty much with every drug that has been approved and is on the market for Parkinson's.
That's amazing. So maybe we could start, Raj, with you telling us a little bit about the process of how something gets from, let's say, an idea in your head to getting on the market. What would the steps take? Maybe you can break that down a little bit for us.
Right. So before anything happens, let's say a compound has to be made in the lab, or you take something that is already available and make it in a different form or a better form. For example, we have carbidopa, levodopa. So doing an extended release, you have that drug, you're changing the formulation and delivery, or getting an inhaled levodopa. So the drug is not different, but the way you are giving the medication for whatever reason is different. So how that medication is going to go to the market versus something that is a brand new molecule, let's say like pimavanserin, which is a medication for Parkinson's psychosis. That medication was developed, then went into initial studies. And we have basically, before a drug actually is taken by a human being, it has to go through a lot of safety, not only in the lab, but also animals. So animals have tried that medication. We have a very good idea of the relative safety of the drug. So it's not the first patient or the first human being taking the drug is what we are going to start learning about it. We know about the medication even before a human takes it. But let's say it has started in the lab, it has been found to be that in the cell cultures or whatever it is, there is a role for it, it is going to be effective. You take it to animals, you look at the safety data there, you feel that there is enough there for the safety. You may actually also look at a disease state in the animal and see, does it make a difference? So not only is the efficacy looked at it, but also some degree of safety is looked at it. So you look at both of them. And then the drug enters a phase 1. So a phase 1 is usually done in normal human beings. So a group of usually young human beings go to a clinical center for research. They get paid to take this medication, and they're given different doses of the drug to see how safe they are, how well it is tolerated. And then the next stage happens in the patients. So then you look at the safety, which is usually a 2A study, where it looks at how safe the drug is in the patients, how well it is tolerated. You may even come up with some potential dose at that time. The next phase usually goes to a 2B phase, where you're looking at, again, in a larger number of patients, getting a better idea about the actual dosing, looking at the safety. And then the phase 3 are what we call the pivotal trials, based on which a drug is approved. And that usually requires 2 large studies for approval, but in certain cases, a drug may get approved based on only 1 trial. So that's the phase 3, which eventually leads to the approval of the drug. After that, once the drug is approved and in the market, we may do post-marketing studies to get a better idea of the safety, how it is used in the clinic. Are there a certain group of patients who do well? And that's phase 4 after it's available. So to give you an idea how long it may take from the time it leaves a lab and animal studies to the time it actually makes it to the point it's getting approved can be up to 10 years. So you're looking at a long time, and certain medications may take even longer. For example, pimavanserin being a brand new compound, I did the first studies back in 1997, and this drug was approved in 2006. So it took close to 20 years to make it to the market. Then you have certain medications that have been approved. Let's talk about a long-acting levodopa, which may take 5 to 6 years to make it in the market. But like I tell people when they want to know, is there some medication I can use? If I'm testing the medication today in the clinic, it is still going to be 3 to 5 years before I can write a prescription for that medication. So it's a long process. It takes a long time to actually make it to the market.
That's pretty amazing. And how, like a clinical trial center like you and I were speaking yesterday, and you said that you're now, how many different trials are you doing?
So right now we are doing about 13 active clinical trials, or we just finished some of them, but about that many.
Okay.
And in different phases of the disease for different reasons, so to speak.
And how does a clinical trial center like you choose which sort of trials that you're going to participate in, let's say?
Right. So for us, because I love doing clinical trials, and that's kind of what I do a lot about. To me, any new medication that is being looked for Parkinson's, which has advantages over what's out there, we are definitely interested in helping out to study that medication. Because the first thing our patients ask us when they come to the clinic, oh, is there any new medication since I saw you last time? And if we go back and look at 10 years from now, there was a huge time period of about 10 to 15 years when there were no new medications. It wasn't like we were not doing trials for it, but we didn't have much success on what things we were testing. On the other hand, if you look at it in the last 4 years or so, there have been about 6 or 7 new medications that have come up. In fact, since COVID hit us in March, there have been 2 new medications that have been approved. So right now, the times are very different as far as Parkinson's is concerned, the medications being looked at for Parkinson's. And the thing is, there are still so many unmet needs in Parkinson's. Parkinson's is a 15, 20, 25, even 30-year progression that we are looking at from that part. So what patients have issues at the beginning of the disease are very different from what's going on 15, 20, 25 years down the road. Some of those things we can help. A lot of those things we still cannot help. So we do need better and new medications from that part.
If you, let's say, since you did mention the unmet needs of Parkinson's patients, and there's a few questions in the chat as well, so we'll maybe hop around a little bit in this discussion, Raj, but I think it'll be stimulating to the group to hear from you, who's a big clinical trialist, sort of what you feel like, and you've been in practice now for, you look so young, but I don't even want to say how long you've been a doctor for, but longer than me. So in your evolution of thinking, because, we all go to medical school, we learn about Parkinson's as a motor disease, right? And the things that we see the most, like tremor and some, walking issues. Now that you've been a doctor for this long, what do you really think are the main things that affect your patients and their quality of life? And where do you see the unmet needs of Parkinson's patients?
So let's start with what is the biggest unmet need in Parkinson's is a cure. You know, or after a cure is even if we can slow the disease down. I mean, to me, those are, so to speak, the biggest thing, because if we can achieve a cure, everything after what I'll talk about is immaterial. Short of getting a cure, if we can slow the disease down, that would also be another big thing that we are able to do. And believe me, we have looked at, I can't even count, over 40, 50, 60 different compounds over the last 30 years trying to find that medication that I could stand in front of you today and say, oh, every patient needs to be on this medication because this definitely slows the clinical progression of the disease. No, we have not yet succeeded with that. Having said that, doesn't mean we have given up. We are still looking at drugs. We have a better idea of Parkinson's. And to me, when people ask us, why have we failed to find that? And a large part of it is we don't know. If I have a Parkinson patient sitting across from me, what caused the disease in that person? If I have 5 people in the room, it's very likely there are 5 different reasons that they had the disease. Maybe one, there was a Parkin gene, one, there was a LAR2 gene, one was some other environmental factor that caused it. But we have, for years, had the theory that, okay, Parkinson's is one disease, and the way the cells die is one way. So if we put a group of Parkinson's patients and throw a medication at them, if we can slow the disease progression, that's what it is going to be, because all the way the cells are dying is the same in every patient with Parkinson's. Well, that's not really the case. So what we have done is used a shotgun approach. We have used medications with different ways of acting, some affecting the mitochondria, some affecting the enzyme MAOB, some affecting the calcium channels. So it's like we have taken different drugs and done trials with it because somewhere in a lab, someone said this has a potential way that the cells are dying in Parkinson's. I'm not saying that's not true, but what I'm saying, if we have 5 patients with 5 different ways the cells are dying, and I use a drug which is only going to help one of the 5, when I look at the data, it's going to be negative. So that has been our one of our big challenges in finding it. If we could say, I have Mr. X, and Mr. X has a LAR2 gene, and LAR2 gene, if we can do something to that, we can slow or cure Parkinson's. That would lead to more personalized medicine, and that's the ultimate goal we have. So to me, cure and slowing progression is by far the unmet need that we have. What about other unmet needs? I mean, I can stay here and just talk to you for an hour just talking about unmet needs in Parkinson's, but think about it. What do we face our challenges in our patients? What do we have very little for? Well, the patients who get dementia, when the memory begins to get impaired, we have very little that we can do about. Parkinson's psychosis, when patients have hallucination and psychosis, well, at least we have one medication in the last 4 years, but before that, we really didn't have anything. When we look at balance issues, usually the Parkinson's medications don't help the balance, doesn't help the freezing. Now, there are patients out there that in spite of trying all the medications, their tremor is not under control. Patients are still having motor fluctuations. That's the on and off after so many drugs that have been approved for it. Patients are still having dyskinesias, the involuntary movements, in spite of so many medications, being looked at it from the dyskinesias. Patients have orthostatic hypotension. I mean, again, we have medications for it, but in spite of that, our patients are getting lightheaded and having syncopal attacks because of that. We have patients who are sleeping all day, excessive daytime sleepiness, constipation. I mean, like I said, I can go on and on, but these are some of the things. We may have one medication, we may even have 7 different medications, but those issues have not been resolved. So we are still trying to find something that would help every patient, not only a subgroup of patients.
So it sounds like you do believe in sort of the more customized sort of precision medicine approach that people like Dr. Espay are talking about. He came on our program when it's talking about the sort of almost like in cancer treatment, there's sort of a need to define in 5 patients what the mechanisms might be for each person, and then kind of come up with you know, a sort of an approach, which sounds really interesting. But I think from a patient perspective, I think there's a lot of a sense that there's many unmet needs, a lot of things that can be very overwhelming. And then if each person needs a different approach, how is any one person really going to feel and get better? So I think, you know, how do you kind of bridge that gap for patients to help them, you know, stay positive and understand sort of where we are with these trials right now, and not lose hope, you know, and sort of how that's going to help them personally.
Right. So I'm actually talking about what happens, why we have not been successful as far as slowing or curing the disease is concerned. Once a patient has symptoms, so whether it's treating their motor symptoms, treating their motor fluctuations, tremor and all that is still one way we are doing it. As far as trying to slow the disease, the thing is our thinking keeps changing. You know, there was a time we all felt MAOB inhibitors were the medication that was going to slow the disease down. Then there was a whole argument whether it's selegiline, whether it's rasagiline. Well, there's not enough data to show it slows the disease down. It may also help symptoms. So how can you say for sure it's not the symptom improvement that we are seeing, but it's the slowing progression that we are seeing? But even though that medication was used in a group of patients, there is some of us who still believe that it has a role in slowing the disease down. I cannot stand here and tell, oh, 100% every neurologist will agree with what I'm saying, because there is data there, but it is not 100% convincing to everyone that it slows the disease progression. Same thing, you know, right now, one of our biggest thing on doing or looking at slowing disease progression is looking at an abnormal protein in the cells, Alpha Synuclein, which causes Lewy bodies in the brain and other cells, which pretty much all Parkinson's patients have. So the thinking behind it is if we can clean or get rid of these abnormal proteins in the cells, then maybe we can slow Parkinson down. So one way to look at it is this abnormal protein happens in every Parkinson patient. Doesn't matter how it began, whether you had gene A, gene B, whatever it was, you still have to have that Lewy body. So if we can clear that Lewy bodies or clear the alpha synuclein in the cells, then maybe we can slow progression down. So right now, I think that is one thing which is being looked at, and maybe that is the way. Maybe a year or 2 years from now, we will be talking about it and say, forget how or what is the actual mechanism. Every patient we know with Parkinson's has, you know, alpha synuclein in their cells. We clean the alpha synuclein, we clean the cells up, and we can make a change in the disease.
Makes sense. Yeah, I mean, it is quite complex, it sounds like. So there's just starting with a few of the questions from the group here. One of the questions is about getting medical devices like DBS approved. You talked a little bit about drug development. Could you tell us a little bit? I know you've done some trials in DBS as well and can speak. So this is the deep brain stimulation devices. Maybe you could speak to getting devices and procedures approved as well.
Right. So deep brain stimulation approval again has changed a lot. So the first approval we worked on was approved back in 1994. That was right after I had finished my fellowship. It was, again, one of the first projects we did. And at that time, the division that approved devices was not that rigorous, because most of the stuff was done for the surgeons. And if a surgeon did a device, it helped them. It used to get approved relatively easily. So the first devices that were approved were much easier to get approved. So when we had the first DBS for tremor or for Parkinson's, which was subthalamic nucleus, globus pallidus, were the targets at that time, all we had to show is this device was used, it was effective, and it was safe. So those things were done. We all know DBS is very effective. We found out it was relatively safe because, you know, you had infections, you had bleeds, you even had a few deaths initially, But again, that was approved based on relatively simple trials, simple studies. However, when the next devices were approved, in other words, the next companies looked at it, they had to do much more controlled studies, trying to have a control group to show that the patient who got the device did better than a control group. And people looked at different control groups. For example, one study was done where one patient after surgery, their device was turned on, and another group where you waited 3 months to turn the device on, and showing that the one who was turned on right away did better than the ones who waited for 3 months. So FDA needed more data from that part. The good part is the insurance companies like Medicare paid for the device because the biggest expense is the device. So it didn't matter if the device was approved or not, as long as it was being studied, the insurance companies were willing to pay for it. But every device that was done had some degree of improvement from the previous device. So like if you look at Medtronic, the contact or the lead that is in the brain has 4 different active contacts. When Abbott went out and did their studies, they went and said, okay, we have 2 contacts which are complete circles, and the middle 3 are partially, like you have 3 additional cuts in it. So it had more programming issues that were going on, or more programming ways you could do it. And then when the next device was looked at, Boston Scientific, they tried to come up, it was rechargeable. So that is their plus that is there. Now, once these devices are approved, when they have to go back and try to change certain things, whether it's a new pulse generator, that can take additional couple of years. So if you look at the Medtronic device, that came out from the idea of when patients had lesions created in the brain, like a thalamotomy, you used electrical impulses. They took the technology from an old pacemaker, and that took roughly 4 or 5 years for approval from the time they started doing the studies. The Abbott one took about 8 to 9 years for doing the studies because they were doing much more control studies. They had to have a control group. The Boston took less because the devices were there. We knew the technology, we knew the safety and all that. So it was looking at actually, can we show the efficacy and safety? So that's what happens with devices. They still have to go through the whole process. With the devices, it is getting to a point that they are trying to put the same standards as a medication. So it's taking longer for newer devices to. And the thing is, just like with medication, for a new medication to come out, there has to be some additional benefit from it. Same thing with devices. If let's say a new company tomorrow wants to come up, there has to be some advantage for them. It can't be another Me Too device that they are going to put out in the market. But to me, the exciting part for the future of DBS and what future trials are exciting about it is, one is lead location, right? So what is the biggest thing in outcome? When people ask me what are the chances of improving? Well, it's really every patient with DBS should improve, except if the leads are not in the right place. So even if it is 3 millimeters away, which you can imagine how little that is, it can make a difference from being 90% better to being only 50% better. So we are looking at software now that can actually show us exactly the location of where the lead is, not only during and after surgery, but also when we are programming. So we know what contact to use and what is the best stimulation parameters to use. And to me, the other exciting thing about DBS is remote programming. One thing COVID has taught us is seeing patients over the internet. What if we could actually program patients over the internet? And I think that is, to me, an exciting future thing that's happening. So it doesn't have to be getting a device approved. Even if we have to do remote programming, FDA also has to approve that, because think about it, there has to be safety involved, that when you're programming over the ultrasound, no one can hack into it and cause issues with it. And also show that it still works, that it is efficacious. So, it's not just the device, it's also some of the other things that go along with it.
That's interesting. There's been a few questions in the chat about side effects and how does a person know when they're going into a trial about what possible side effects they may encounter, and also when a drug or a medication or a device is studied, how do we know what the side effects are from, you know, the actual medication versus, you know, so can you tell us a little bit about, you know, the randomized control sort of trials and that kind of thing? So maybe speak to the counseling about, you know, when you're entering a study about the side effects, and then also maybe how we know with trials and the type of trial you do, what we attribute as a side effect to the actual drug or procedure.
Right. So first of all, with every next phase of the study, we know more about the potential side effects that can happen. Having said that, we know a lot about the potential side effects when a patient is getting into a phase 3 study. Having said that, I won't say that a drug that was safe in phase 1, phase 2, we found certain risk in phase 3 for which a study was stopped. No, we have had those things happen too. And that's why a phase 1 or actually a 2A study may have only 25 to 30 patients, a phase 2B may have 100 patients, and a phase 3 may have 300-400 patients. But when a drug comes in the market, it's hundreds of patients or thousands of patients who are taking the drug. So the more patients who take the drug, the rare side effects only show up at that time. The common side effects, you know, you will see it with a small group, but the rare side effects happen with the larger group. Fortunately, most of the rare side effects are not something that is going to stop the trial or be a big safety concern. But at times, a rare side effect could be a decision has to be made whether it's safe enough to continue it. Having said that, like I said, the first thing happens in normal volunteers. So the first study, a patient gets the drug, very high doses, not a patient, but a control group before any patient gets the drug. So we get an idea about, oh, this person had nausea, this patient had vomiting, this patient was confused, whatever it might be. So from that, we get some idea. So when we do it in the patient, we provide all the potential side effects that occur to the next group, which is the patient, that this is what was seen in this group of normal volunteers. So this is the potential side effects. When the second group goes through it, you have a little bit better idea of the side effects. The number, the frequency might increase. So when the next group comes, we will actually have a better idea of telling a person, well, the nausea, you have a 10% risk of it. Sleepiness, you have a 12% risk of it, et cetera, et cetera. So you already have a reasonable idea by the time a patient is going to be started on that medication. The other thing is, as the study is going on, anything that was not in the original consent form, if it is seen. So let's say this is a trial which is in 20 centers, and my center, everyone is doing well, but let's say a center in California, they have a side effect that was not reported in the consent form. I would be informed of that immediately. I would have to inform all my patients who are in the study, oh, by the way, this patient had so-and-so potential side effect, and they have to continue, okay, one person got it, it's fine, I want to continue. Or they may decide, well, I want to stop, I want out of it because I'm concerned about this side effect. So it's not just when you enter, you're told about the side effect. Any ongoing side effect that was not part of the consent form, you update it and do it. The other thing is, when we are doing a study, when we see a patient come in, whether they are on an active drug or whether they are on a placebo, anything that's new, it comes down as a side effect. So let's say a patient is in a trial and they have an unrelated fall and fracture their arm. Well, you know, that wasn't the drug doing it, but we are not 100% sure. So we would still report it as a fall that occurred or a fracture that occurred as a side effect. So at the end of the study, you know, we have two groups, one who got the drug, one who got the placebo, and then we put the side effects down, because some of these side effects happen in the placebo group too, patients who got a sugar pill. So that's why when people ask me, oh, does this drug have side effect? Well, every drug has side effects, because we know even patients who get placebo, who get a sugar pill, they also complain of side effects. And, you know, I like to look at it and see how much side effect a person is having. Let's say an active group had 10% had nausea, placebo group had 5%, and I look at it, it's okay, 5% risk of nausea. Now, some people may say, well, it's 10%, forget the placebo group, which is fine too. But always, any side effect we look at, we don't know if a patient is taking a drug or not. We don't know if it's their disease causing it, it's the medication causing it, it's the flu they had. All of those are reported as potential side effects.
And I know, Raj, that you've been on some data safety monitoring boards as well on trials. Could you tell us about what that is and what the role that you've played on?
Right. So the data safety monitoring is basically any trial that is being done. Usually, the FDA will say they need a data safety monitoring. The IRB, which approves the trial at that institution, will also insist on having a data safety monitoring committee. And the data safety monitoring committee usually involves a group of experts, which usually would be a neurologist if it's, let's say it's a Parkinson study. So a neurologist with experience in Parkinson's, usually a statistician, depending upon certain risks that the drug may have. Let's say there's a potential cardiac risk. They may have a cardiologist. If it's a surgical trial, they may have a surgeon in there. So they usually have about 5 to 6 people on a safety committee, and their job is to look at the side effects as they are coming in. They especially look at any serious side effects. And by serious side effects, we talk about patient going in the hospital, patients requiring surgery, patients, depending on the study, some of it may also require, if they go to the ER, it may require anyone getting pregnant. So there are certain criteria where we call it a serious side effect. So when a serious side effect occurs, the investigator has to make a decision, was it related to the medication or was it unrelated to the medication? But when the safety committee looks at it, they take into consideration what the investigator said, they look at the AEs that are occurring, and then they may ask for more data. They may ask, oh, have you done this test? And then the investigator has to go back and do it and provide it. So they are like the person who's monitoring the safety of the study to make sure they can go through with it. A lot of data safety committees have it in it that if, let's say, there's a certain safety that's too high, they could either temporarily stop the study, look at it, why it is happening, or they may actually stop the study completely. So they have that power to do a lot of things as far as when they're monitoring the study. Some of the data safety, they may have pre-endpoints, like at a certain point in the study, they would look for the efficacy and the side effect. And let's say if the efficacy is so powerful on the group, on one group versus the other, because they don't know which group is getting the drug or which is getting the placebo, they can ask for that information. They may say, okay, this group, there's such a big difference between the patients who are getting the drug. I think that you don't need to continue it. And that could also happen. So not only are they checking the safety, they may also be looking at, it is not that the people who are getting placebo are getting denied of a medication that would be beneficial for them. So the part of the data safety is very important. So anyone getting into clinical or in a clinical study should ask, is there a data safety committee who's actually monitoring the safety on the study?
Okay. Could you also teach us a little bit about you mentioned the IRB committee. Could you tell us what that is? And, you know, sometimes it's in the local institution, there's also a central one. Sometimes could you explain the difference?
Right. So before, first of all, if someone needs to do a large trial, so if I have a medication, I think it's helpful in Parkinson's, I'm going to do this wonderful large study. I usually get a group of experts together who sit down, come up with the study design, the protocol, etc. Once that is done, then you go to the FDA and you show it to the FDA and say, this is a drug we feel helps in certain symptoms of Parkinson's. We want to study this drug. This is how we propose studying it. So the FDA, from their standpoint, would look at the protocol, look at the safety, and then come back with recommendations. Well, needs to be done, that needs to be done before we'll approve it, or we don't like this, you need to change it. So they have a discussion with the FDA before they can actually start the study, and the FDA has to give approval. Okay, we both agree on this, go ahead and do a trial with whatever, 50 patients in 5 sites or whatever it might be. Once that's approved, they go to the centers. So when you go to the centers, some centers have their own institutional IRB. And what the job of the IRB is to look at the protocol, look at the study, look at the safety, and make an independent decision that yes, it is safe. They have to look at the consent form, which the patient signed, and the consent form has to be in a 8th grade language. In other words, a person in 8th grade can understand the consent form. So they do all that. If again, they want to make changes, they are completely free to recommend changes. Usually when it's an FDA study, they are paying more attention to the consent form, that the person reading the consent form understands and approves it. And again, on the IRB, there are a group of people there who are making this decision. And this group usually involves not only physicians, but it also involves some patient advocates on it. It may involve some legal people on it. It may involve statisticians on it. So all of these people are looking at it and as a group voting that, okay, this study is safe, it has benefits on efficacy, the protocol is well written, and they will approve it. The difference between an institutional IRB and what we call a central IRB is a central IRB, basically once it approves it, the number of institutions underneath it already also have it approved. In other words, instead of going to 5 different IRBs, one IRB can approve it and all the 5 institutions then get an approval for it. So it really makes it much easier for the clinical center to do it because they're working with one IRB who's taking care of all the 5 centers rather than each center individually going to it, each center coming up with a different consent form and different changes.
Okay, that makes sense.
So from a patient standpoint, it does not matter whether it's a central IRB or a local IRB. They both have to be certified and have to be people or IRBs that are doing the job that they are doing.
Okay. Maybe you could teach us, there are some questions specifically about certain medications, and we'll get to that hopefully, but I wanted to just have you have a chance to help the patients understand how they can get more involved in clinical trials. If you have a sense that COVID has affected this, you know, maybe you could teach us a little bit about what your guys are doing differently, because I know you do a ton of trials, and how you kind of are working around some of these issues with COVID.
So, you know, clinical trials are very important because that's how we get new medications approved. That's how we know what medications are going to work and how safe these new medications are. And if we look overall, maybe 5 at the most, 10% of our patients are interested and willing to do trials. And the reasons could be multiple. You know, maybe they don't have the transportation. Fortunately, most of the new trials have places where they may allow you to get a taxi in or a transportation in. So always ask the center, if you're interested in doing a trial, I would love to be part of it. However, I don't have a way to come here, or both I and my spouse don't drive. And most of the centers will figure out a way from the sponsor to get information or funding for the transportation part. But I think, why do patients do studies? Well, you know, there are multiple reasons. Some patients are definitely wanting something better because whatever they are taking is not helping them. And they don't want to go for, let's say, DBS, they're having off time. So they want to do a new study. And often the studies, if there is a drug which is helpful, the sponsor will give them the free medication until it comes to the market. So not only they participate in the study, but after that, they get the free medication for maybe 2, 3, 4 years, as long as it is helping them. So that's another advantage. Then there are certain patients who just want to do it for the science of it. You know, I have Parkinson's, I want to help get the next generation medication or whatever it is, and be part of it. And then there are people who want to know how these studies are being done. And for them, being part of it gives them an exact reason on how this is done. So there are multiple reasons to do it, but unless we have patients volunteering for the studies, it's difficult to do these studies and get approval from new medications. So I think that's an important part to remember. Your question about has COVID affected us? Well, personally, I'm in Kansas, and we were completely closed down for about 2 or 3 weeks. And after that, we gradually opened, and our research patients, we were able to see them even during the initial COVID crisis. So for us, as of today, our research, our clinical trials are no different than they used to be. But having said that, there are patients who still don't want to come in for a clinical trial because of concerns of COVID. On the other hand, there are places, let's say, for example, I don't know about California, New York, where they still don't allow patients to come to the clinic. So those patients or those clinical trials may either be on hold, but a lot of these places have opened it up as telemedicine trials. So people are doing their visits over telemedicine, which can be done to a large extent. But again, there are certain drugs, for example, we have intravenously that are given. So those can't really be given over telemedicine. And that's another thing that has to be given in the clinic. So there are ways that we are getting across it. Is it affecting the recruitment? Yes, it is definitely affecting recruitment of patients. Is it affecting that how long that study that was being done in, let's say, 6 months now may end up taking us 8 to 9 months? Yes, it is definitely possible. There are clinical trials that have been put on hold. We had a trial that we were going to start this month. Now it is on hold until next year, basically because they want this crisis to be over and they can do. So yeah, has it affected? It's definitely affected the trials. And it's definitely going to delay approval of new medications that we are going to have. But still, a lot of sites are still doing it, and we are trying to keep it going.
So there's been a question or two about how patients can find out more, because I think some of our patients live in rural areas, maybe not so close to centers. Do you have resources, Raj, about where people can find out about if there are remote trials or telemedicine trials exclusively? Because I think that may expand the reach of people being able to participate.
So all the clinical trials that are ongoing are listed on a website called clinicaltrials.gov. And this includes any trial, not just Parkinson's, any trial out there that is there. So I strongly recommend people may want to go to clinicaltrials.gov. And what they can do is put up the disease. So let's say Parkinson's disease. They could put up what area they are in. So here, like say Kansas, and then it'll list all the trials that are there and how far you are from each site. It would also give you what the study is about. So is it early disease? Is it in patients with off time? Who the sponsor is? What all do you have to be part of the trial? So that's one way to definitely do it. Another way to do it, to go to the Michael J. Fox Clinical Trial Finder, and you can go there and register. And if there's any study that kind of goes along with what your qualifications are, let's say you're looking for a study that helps the tremor, and if such a study comes on, they will inform you about being part of it. So to me, those would be a couple of good resources to look for and sign up and be part of trials from that part.
Okay, that sounds really good. There's been a few questions about levodopa preparations here. One is about subcutaneous levodopa preparations in trials, and also about duopa, and if they would be possibly going to extend duopa studies to do a 24-hour dosing. Do you know anything about those two questions, Raj?
Yeah, so as you know, duopa is a levodopa gel where a tube is placed through the stomach into the jejunum, and it provides levodopa continuously in the jejunum. And again, this is for patients who are having on and off, and it provides a much more level levodopa level, so the patients are not fluctuating or having dyskinesias. As you know, this preparation is not available for 24 hours in US. Having said it's not approved for use, if there are patients who need it for 24 hours, most of the insurance companies will approve it and people can get it for 24 hours. There are no large trials being considered or being performed for 24 hours of DOPA therapy that I'm aware of. Well, what the next stage is, which is 24 hours, and if you look at it, there are two subcutaneous levodopa preparations that are being looked at. One is by AbbVie, which is called AbbVie 951, and the other one is with Neuroderm, which is ND0661. They still have numbers, they don't have names yet. So for example, the AbbVie, it's a subcutaneous, so you don't have to create a lesion to put a tube in. So it's kind of like a diabetic pump, except the pump is a little bit larger, and it provides continuous therapy for 24 hours is what it does. Again, people who are interested in it can go and register or look for sites on clinicaltrials.gov. But both Neuroderm and AbbVie are doing subcutaneous infusion. There's also a long-acting oral preparation by Amneal, which is called IPX. I think it's IPX203, sorry. IPX203, which is 3 times a day extended release capsules. So it's not a tube, it's not surgery, it's not subcutaneous, it's a capsule. And again, patients can look up on the clinical trials website under IPX and see if there's a center close by. And both these, the subcutaneous levodopa and the long-acting levodopa, are for patients who are having off time. So that would be what would get them in this study.
Okay. And what's going on with the apomorphine pump trials in the US? Could you update us on those?
Right. So as you know, apomorphine injections have been approved and available in the United States for, I think it's about 14 years or so now. So it's been available a long time. And in Europe, they have been using a pump and providing the subcutaneous infusion continuously. The thing is, they were doing it in Europe without controlled studies. They had a lot of experience there, and the European Union basically approved apomorphine pump based on the studies that were there. For it to be approved by the FDA, those studies were not adequate. So they had to do a controlled study to get approval in the United States. That has been submitted to the FDA, the data. The data is positive. The safety is there. One of the things is being that the drug, which was, with one company was sold to another company, and that at times also takes a little time off. But we are very hopeful that the pump should be available sometime next year because, like I said, it's with the FDA, and FDA is looking at the data. And once they approve it, it should be in the market and available in the United States.
Okay, that's good to know. And then we have about 8 minutes left, Raj. I wanted to give you some time to talk a little bit about what you think are the exciting areas of, so we talked a little bit about the levodopa therapies, and you talked about these sort of longer acting ones. You talked a little bit about the DBS and your excitement with using telemedicine to program, and also some of these novel approaches with DBS. If you wanted to talk about any of those, that's fine too. But even in the non-motor realm and other sort of exciting areas that you think are really sort of the, if you were a patient, you'd be the most excited about, and what people should really be staying tuned for.
Yeah, so, it's like if you have a kid, you have a young kid and you have, let's say, 13 kids, and you say, who are you most excited about? It's kind of tough to say, or asking me who's gonna go finish high school and who all are gonna drop out, it's difficult to say. So we are at that stage. We have, 13 kids who are just entering school. How many are gonna pass high school and be available commercially, and how many are gonna drop out at 2nd grade, 4th grade, 5th grade? Is kind of difficult to say. So right now, all of that is exciting, and everything we do is exciting to get it. And, like I said earlier, our unmet needs start with slowing disease progression. And right now we are looking at medications that clear the abnormal protein Alpha Synuclein. We are looking at reducing the inflammation related to Alpha Synuclein and trying to do some neuroprotection in the cells to help with it. So there are a few compounds looked at very early, and 2 of them are antibodies against alpha synuclein, where the antibody will bind to the alpha synuclein and basically, cause the protein to be disintegrated, so to speak. So the two of those are really further along. One is by a company, Roche, called Prasinozumb, and another one is Sinapanabab. So they are very tongue twisters here, and I'm sure I'm completely torturing them because the names have just come up. So I try to remember right now by saying one is a Roche compound and one is a Biogen compound. And both are in early studies, but their goals are very similar and the way they work is very similar. Then you have another company, Sun Pharma, which is looking at the tyrosine kinase. And what it is supposed to do, again, is cut down on the alpha synuclein in the brain and the inflammation along with it. But it is not using antibodies, it's using a different mechanism. But the idea, again, being trying to clear the alpha synuclein. Another group of drugs that is out there is what we call GLP-1R activators or agonists. These are similar to drugs that are used in diabetes, but we also have seen that these drugs actually help with neuroprotection, reduce the inflammation in the brain. And Neurally is a company that is, again, looking at it in an early disease state. So to me, these are exciting because we are looking at trying to slow the disease progression. I'm saying it's so early that even though I'm excited about it, I can't tell you for sure one of this is going to be the big breaker out there. But we are looking at drugs to slow progression. But the other thing that we didn't talk about are gene therapies. And to me, what is gene therapy is that you basically surgically implant certain genes in the brain, which helps production of dopamine, which is one of the main neurotransmitters that is affected. The thing is, some of the early studies with open label, in other words, everyone getting the gene, like I said, the phase 2A study was very positive, but you need to do controlled studies to get a little bit better idea on. So one of them is called V as in Victor, Y, AADC gene therapy. That's a little further on. And it is right now doing a control study. So one group gets the drug, one is a sham surgery. And the thing with the gene therapies is hoping to preserve these cells longer that are being produced dopamine. And again, with the gene therapies, you need longer studies because you're not going to have those answers in 2 months where you could get with DBS. These take a year or 2 years for us to know. Again, these are being done in a little bit more advanced patients who are having motor fluctuations. So again, if people have off time, they are interested in a gene therapy, that would be one of them to consider. Another one is a gene therapy called Axolenti, Lenti, and that has 3 genes in it. And again, with the idea to increase the dopamine production, and it's by a company called Axovent. So those, again, are trying to have people or have the body make more dopamine themselves rather than trying to give a drug or whatever it might be to create dopamine. So to me, that's a different mechanism that's very exciting because often people ask, when is stem cell going to be available? And stem cell is still pretty far. I doubt I would be still working when they do the large clinical trials related to stem cell, because right now there's one small 10-person study going on in Australia. So it's a very small study. We still need a lot of safety data. The other thing with stem cell comes out is what kind of stem cell one is being used. So there's still a lot of challenges with stem cell. But just because we still have a lot of hope for stem cell doesn't mean we stop looking at what is right now available and using what is available for us right now. So that's an important part to remember. There are a few studies we are doing with dementia and Parkinson's with the company Eli Lilly. And again, the drug doesn't have a name. It has a number LY3154207. So I know the numbers and this no one is going to remember. But again, the exciting part is we are studying for dementia. Same way, there's a very exciting drug we are studying for orthostatic hypotension. We are studying a drug for excessive daytime sleepiness. All these are drugs out there. A lot is going on. It's a great time to find something for Parkinson's. And I'm hoping we will have some of these which will graduate with honors.
I like that analogy that you have of the children. We just started the conversation with one child going to medical school, and now we're having children dropping out in grade 2. So we've really come a long way. Well, I think this is really interesting, Raj, to just hear your knowledge, because you've really been sort of alongside cheering for so many of these medications and devices as they've been studied, and you have systematically been very good about creating an idea and then figuring out a way to systematically get it from, just that idea to really a trial. So I applaud you for your hard work, and it takes systematic ability to keep organized and think in a way of how to learn from the previous failures to get, to some success. So I applaud you, and I appreciate all the mentorship that you've given me. And we as a community, I think of folks who care about Parkinson's, applaud you for, giving us this wealth of information so that we can hopefully learn from these mistakes and move forward in a great way. So it's exciting that you think that there's excitement out there to be had, and that you think there's a lot of hope for new developments currently, because that's great to hear. So thanks so much for your time, and appreciate you so much, and great to see you, even if it's virtually, Raj. I'll hand it back to you, Rebecca, for our final wave.
Yes, so thank you so much. This was amazing. What a great presentation, and so much hope for what could be in the future. So thank you so much for your time. If everyone can turn their cameras on, we're going to end this with our PMD Alliance wave. Thank you all. Have a wonderful weekend, and we will see you on Monday.
Bye.
Bye.
Thanks, Raj.